Hi student, today you will study one of cardiovascular system. Cardiovascular system refers to a closed system of the heart and blood vessels. The heart pumps blood vessels allow blood to circulate to all parts of the body. The function of the cardiovascular system is to deliver oxygen and nutrients to cells and tissues, and also remove carbon dioxide and other waste products from cells and tissues. When you hear about cardiovascular system, what do you think first? Probably heart. What is going in and out of the heart? Blood? Can blood freely flow inside the body without conducting tool? No. Blood needs blood vessels. So, heart, blood, and blood vessels consist of cardiovascular system, and today you will study blood. Blood is the fluid tissue in the human body, and blood is a type of connective tissue. Blood consists of cells and cell fragments surrounded by a liquid matrix. Let's look at the function of blood. Blood transports gases such as oxygen and carbon dioxide, also transport nutrients from the digestive tract and waste products to the kidney for elimination. Blood carries many of the hormones that regulate body processes from one part of the body to another part. The blood cells make up an important part of the immune system, which protect against foreign substances such as microorganisms and toxic molecules. Blood also involved with body temperature regulation because warm blood is transported from the interior to the surface of the body. On the surface, heat is released from the blood. The average blood volume of males are about 5 to 6 liters. In case of females, 4 or 5 liters. Blood has a cellular component and liquid portion. Cellular components are composed of blood cells such as red blood cells, white blood cells, and cell fragments called platelets are one of cellular components also. These cellular components are called formed element. Formed element. The liquid portion of the blood is the plasma. So this plasma is extracellular matrix to blood cells. If you spin blood in a centrifuge, the heavy formed elements are packed down and plasma, the liquid portion of blood, plasma rises to the top. So you can see here formed element on the bottom of the tube, and then top portion of the tube, you can see liquid portion of blood, plasma. When you look at this bottom portion, from a formed element, the reddish mass of the bottom of the tube are erythrocyte. Between erythrocyte and plasma, there is a thin whitish layer. This is called buffy coat. The buffy coat is at the junction between red blood cell and plasma. It is barely visible. This layer contains the remaining formed element such as white blood cells and platelet. Hematocrit refers to the ratio of the volume of red blood cell to the total volume of blood. So, hematocrit is about 45%. This means erythrocytes account for about 45% of the total volume of a blood sample. white blood cells and platelets in the buffy coat contribute less than 1%, and plasma makes up most of the remaining 55% of whole blood. So 45% of whole blood is filled with erythrocyte. We express that hematocrit is 45%. Let's think about the liquid portion of blood first. Plasma is a pale yellow fluid. Plasma consists of approximately 90% of water, 70% of protein, and 2 or 3% of other substances such as salt, nutrient, respiratory gases, and waste product such as urea or ammonium. Plasma proteins include albumin, globulin, and fibrinogen. Albumin, do you know where can you find albumin outside easily? When you crack the egg, the white portion of the egg composed of albumin. In blood, albumin makes up 58% of the plasma protein. And these albumin regulate osmotic pressure. In other words, the more albumin, the less water loss occurs from the blood. So, albumin prevents water from diffusing out of a blood vessel. Globulins occupies about 38% of plasma protein. Some globulins, such as antibodies, are part of immune system. So globulins protect the body from pathogen. Also, globulin proteins are transporting ions or copper. Fibrinogen constitute about 4 to 5% of plasma protein. Fibrinogen is involved in chemical reaction for blood clotting. If fibrinogen converts into fibrin is like a thread-like protein, then blood clot forms. So fibrinogen is a clotting factor. Let's solve this question. Which of these is not a component of plasma? Oxygen, present. Sodium ions from salt, present. Water, present. Urea, waste product, present. What about platelet? Platelet is present in buffy coat layer, not plasma. So the answer is C. When you look at this right panel, you can see micrographic view of a formed element. So formed element, what kind of blood cells belong to formed element? This is easy way to memorize. You just take out the first letter from this sentence. Never let monkey eat bananas. Never N represent neutrophil. Let L lymphocyte. M monocyte. E eosinophil. B basophil. So this is the easy way to remember the formed element. When you look at this micrographic view, you can see this round, small, pink structure represent red blood cell. You can see vast number of red blood cell from this view. And you can also see the cell size is larger than red blood cell. These are all white blood cell. And you can also see some small dot. These are platelets. In addition to this sentence, you have one more important formed element. That is erythrocyte. We will study red blood cell first. This is the structure of a red blood cell. The diameter of red blood cell is about 7.5 micrometer. And red blood cell has biconcave disc structure. This structure increases surface area by 30% for the purpose of rapid diffusion of oxygen. Red blood cell does not have nucleus nor organelles either. Therefore, red blood cells are not regarded as a true cell. So ordinary true cell should have nucleus, but erythrocyte does not have a nucleus, so they are not regarded as a true cell. It is a cell, but it's not true cell. The cytoplasm of red blood cell is filled with hemoglobin. About 97% of red blood cells are filled with hemoglobin. This is the structure of hemoglobin. Hemoglobin consists of 4 polypeptide chains and 4 heme groups. So here's one polypeptide group and exactly identical polypeptide group here, second, third, and fourth. 4 identical polypeptide chains associate together to form hemoglobin. And each polypeptide group contains heme group. And each polypeptide chain is called globin. Globin. And each polypeptide group also have heme group. So heme group with globin, we call them hemoglobin. Each heme group is a red pigment molecule containing one iron atom. One oxygen molecule becomes associated with each heme group. When most of the hemes are carrying oxygen, blood has a bright red appearance. Since red blood cell does not have mitochondria, oxygen picked up from the lung capillary is not consumed, but oxygen can be conserved until released to tissue capillaries. In ordinary cell has nucleus and mitochondria. This ordinary cell receive oxygen, then that oxygen enter mitochondria for cellular respiration. But red blood cell does not have mitochondria. Erythrocyte does not contain any subcellular organelle. It's almost 97% is occupied with hemoglobin. And this hemoglobin, inside the hemoglobin, there's a heme group. This heme group binds with oxygen, and this oxygen is not consumed because there's no mitochondria in red blood cell. So just oxygen bind to heme group and flow through bloodstream and deliver oxygen to tissue cell or capillary blood. The lifespan of a red blood cell is about 100 up to 120 days, and red blood cell originate from bone marrow. Another type of formed element, white blood cell. White blood cells lack of hemoglobin. But leukocytes have a nucleus and organelle. Therefore, they are regarded as complete cell. They are true cell. Although white blood cells are component of the blood serves primarily as a skillful means to transport these white blood cells to other tissues. Why? white blood cell can leave the blood and move by amoeboid movement through the tissue. And in the tissue, especially in loose connective tissue, they have a function to remove dead cells, debris by phagocytosis. So when white blood cells leave the blood and move into the tissue is called diapedesis. Diapedesis is the process of circulating leukocyte which left capillaries. What does leukocyte do once they move from blood vessel to tissue? The function of a white blood cell is to protect the body against invading microorganisms and remove dead cells, debris from the tissue by phagocytosis. White blood cells originate in bone marrow. There are about 4,000 to 11,000 leukocytes present in cubic millimeters of blood. Leukocytes further categorized into two types. One is granulocyte, the other one is agranulocyte. Both granulocyte and agranulocyte have a nucleus. So, as I said before, white blood cells are true cells. The main difference between granulocyte and agranulocyte is the presence or absence of granules. Neutrophil, eosinophil, basophil belong to granulocyte, whereas lymphocyte and monocyte do not have cytoplasmic granule. Therefore, they belong to agranulocyte. A means no granule. This is microscopic view of leukocyte. All leukocyte has nucleus or a nuclei in a multi-lobed or bilobed shape. When you look at granulocyte, these are the nucleus. So there are 3 nucleus. So we call this multi-lobed nucleus. Here's a bilobed nucleus. And you cannot see nucleus clearly from basophil, but here's S shape or U shape. So there is a bilobed nucleus. In terms of agranulocyte, they also have a nucleus. Lymphocyte has large nucleus, and monocyte has kidney bean shaped nucleus. Granulocyte has granule in the cytoplasm. Here's nucleus. The rest part you can see some granule in the cytoplasm. And those granules stained in red in terms of eosinophil. In neutrophil is stained with pink or purple, and basophil is stained with purple color. Agranulocyte lack visible cytoplasmic granule. There is no granule in the cytoplasm. This image is not very clear, but when you observe on the microscope, you can practically understand, but you'd better practice yourself and get used to and identify when you look at just this image, you need to know what is the name of this leukocyte. Okay, let's look at each leukocyte one by one. First, neutrophil. Neutrophils are the most common type of white blood cell, and neutrophils have small cytoplasmic granules. Here's small cytoplasmic granules. Their nuclei are commonly lobed. Can you see? This one we call that lobed. The number of lobes varies from 2 to 6. In this panel, you can see 3 lobed nuclei, and in this neutrophil, you can see maybe 1, 2, 3, 4, 5 lobed nucleus. Neutrophils usually remain in the blood for a short time, then move into other tissue. And in the tissue, neutrophils phagocytize microorganisms and other foreign substances. Neutrophils also secrete a class of enzyme called lysozyme, which are capable of destroying certain bacteria. When you have inflammation, and then when you cured from inflammation, later you might see some pus from inflammation site. This pus is an accumulation of dead neutrophil, cell debris, and fluid at site of infection. There are 3 to 7,000 neutrophils in a cubic millimeters of blood. When you have higher number of neutrophil reflex, there is some infection occurs in the body. Eosinophil. Eosinophil compose 1 to 4% of all white blood cells. Granules are large and stained in red. These are the granules and stained in red color. Cytoplasmic granules of eosinophils stain bright red with eosine. Eosine is an acidic stain dye. The stain dye name is eosine. Therefore, since this blood cell is stained with this dye, therefore it has name eosinophils. Nucleus has 2 lobes. In 2 lobes, interconnected by a broad band. Granules of eosinophil contain digestive enzyme during allergic reaction and parasitic infections. So their numbers are elevated in people with allergies. In spring, some people have a sensitive allergy to pollen powder. So at the time, eosinophil secrete some enzyme during allergic reaction. Chemicals released by eosinophils are involved with the destruction of a certain worm parasite also. So eosinophils play a role in ending allergic reaction by phagocytizing allergens. There are 100 up to 400 eosinophils in a cubic millimeters of blood. So eosinophil represent to allergic reaction. Basophils. Basophils compose 0.5% of all white blood cells. So it's not many. Nucleus usually have U shape or S shape. It's hard to see the nucleus of a basophil, but you can imagine basophil has 2 lobes, and granules secrete histamine. Histamine helps initiate inflammation and acute allergic reactions. So the function of basophil in inflammation mediation. And usually the later stage of inflammation can be taken care of basophil. So early allergic reaction can be reacted by eosinophil, but later stage of inflammation can be taken care of by basophil. Basophils are the very rare among formed element, and is hard to find from blood sample under light microscope. And usually granules hide nucleus shape, and the number of basophils are not many in white blood cells, about 20 to 50 basophils in a cubic millimeter of blood. Agranulocyte. One of agranulocyte is lymphocyte. Lymphocyte consists of about 20 to 45% of white blood cells. So there are 1,500 to 3,000 lymphocytes in a cubic millimeter of blood. Dark purple blue nucleus, it's a big nucleus, and then outside the nucleus you might see outline. Pale blue cytoplasm appears as thin rim of pale blue cytoplasm. The most important function of a lymphocyte is immune system. Two main classes of lymphocytes are B lymphocyte and T lymphocyte. B lymphocyte produce antibody. T lymphocyte involved in directly fighting foreign invaders. So let's look at in detail. Two main classes of lymphocytes are B cell and T cell. B cell, B lymphocyte produce antibodies. B lymphocyte multiply to become plasma cell. These plasma cells secrete antibody. Antibodies bind to antigen. So antigen coated with antibody. This antibody-coated antigen can be easily detected by macrophage. Macrophage actively commit phagocytosis. Therefore, B lymphocyte facilitate macrophage detect antibody-coated antigen. T lymphocyte is called killer cell or T cell. Why T lymphocyte is called killer cell? Because T cell directly attack foreign cells by binding to antigen-bearing cell. This cell, orange color cell, infected by foreign cell. Then orange cell produce antigen and then put antigen on the membrane. It's like a flag or a tag. This antigen, this flag antigen can be recognized by T lymphocyte. Then T lymphocyte bind to this antigen of infected cell. Now antigen, antibody from T lymphocyte form complex. Then T cell make a hole, punch membrane and make a hole. Therefore, this cell become die. In other words, T cell trigger apoptosis. As a result, infected cell become die. Another type of agranulocyte is monocyte. Monocyte compose 4 to 8% of white blood cells. So there are 100 to 700 monocytes per cubic millimeter of blood. Monocyte is the largest of white blood cell. The size of white blood cell, when you consider the size of white blood cell, monocyte is the largest white blood cell. When you look at this figure, you see dark purple nucleus like a kidney shape. After monocyte leave the blood and enter tissue, monocyte enlarge and transform into macrophage. So, when this blood cell flow inside the blood vessel, it is named monocyte. This monocyte squeeze out of the blood cell and present in the tissue, we call them macrophage. Macrophage phagocytize bacteria, dead cell, any other debris within the tissue by forming pseudopod. So monocyte has a capacity to move amoeboid movement to form pseudopod. Therefore, using pseudopod, macrophage get close to the bacteria, dead cell, any other debris, and then ingest them. It's very important in fighting chronic infections. The last one in the formed element is platelet. Platelets are cell fragments generated by breaking off from megakaryocyte. Here's megakaryocyte cell, and cytoplasmic portion of a megakaryocyte split off, and each split off they form platelet. So small amount of cytoplasm enclosed by membrane. This cell fragment is surrounded by plasma membrane and a small amount of cytoplasms are inside. Once pieces of cytoplasm split off and they become cell fragment, then they enter bloodstream as platelet. When you look at this panel, platelet is really small. Think about that. It is just a small piece of cytoplasm from megakaryocyte. Therefore, the size of a platelet is really small. It appears as a small dot. About 300,000 platelets is considered a normal number of platelets for cubic millimeter of blood. Platelet release chemical necessary for blood clotting. Therefore, platelet help seal off large wound in the vessel. So platelet adhere to collagen near the edge of a tear in blood vessel. When you have a scar, blood vessel is torn, and near the edge of a torn in blood vessel, and platelet adhere together with the collagen and form blood clotting. So what could be the answer? Platelets are derived from megakaryocyte. Platelets are cell fragment. Platelets play an important role in clot formation. Therefore, the answer is D, all of them. How can blood cell formed? The process of blood cell production is called hematopoiesis. In the fetus, hematopoiesis occurs in the liver, thymus, spleen, lymph node, and red bone marrow. After birth, hematopoiesis is confined primarily to red bone marrow. Here's bone and bone marrow located within all bones. And there are two kinds of bone marrow, red bone marrow and yellow bone marrow. And red bone marrow actively generate new blood cell. Red bone marrow contain immature red blood cell, and red bone marrow is located between trabecula of sponge bone of axial skeletal bone. So here's trabecula region. So here is sponge bone, and inside the sponge bone there's trabecula, and that is the portion red blood cell can be produced. And also you can find red bone marrow in iliac crest or proximal epiphyses of humerus and femur. Your arm or your leg, you have long bone, and inside the long bone, close to the end of long bone, there is epiphysis plate, and that proximal epiphysis plate, you can see sponge bone, and that is the area where blood cell produce. Yellow bone marrow is dormant. It contains fat cell, so it appears yellow color. But yellow bone marrow make blood cells only in emergency. So this sponge bone is magnified, and then you can see trabecula of sponge bone. And you can see immature blood cells are formed. So bone marrow as the site of hematopoiesis. So you can see immature blood cells, and blood cells keep generating, and mature blood cells enter the bloodstream through endothelial cells of sinusoid. You can see here's blood sinusoid. And once blood cell mature, then they enter the blood vessel by way of blood sinusoid. All the formed elements of blood are derived from a single population of cell called stem cell. The name of stem cell is hematopoietic stem cell. Hematopoietic stem cell. These hematopoietic stem cells continuously differentiate to give rise to different cell lines. Each cell line ends with the formed element of a particular type. Let's look at this diagram showing cell lines in blood cell formation. All formed elements originate from one cell type called hematopoietic stem cell. Hematopoietic stem cell is pluripotent. In other words, it has enormous power and potentiality to differentiate into different kinds of cells. Hematopoietic stem cells divide continuously and make two types of progenitor cells. One is myeloid stem cell and the other is lymphoid stem cell. Lymphoid stem cell produce lymphocyte. And myeloid stem cell produce all the other formed elements such as erythrocyte, leukocyte, and monocyte, platelet. Myeloid stem cell produce all other formed elements except lymphocyte. So erythrocyte, leukocyte, monocyte, megakaryocyte are generated from myeloid stem cell line. Let's look at red blood cell. This is the process of genesis of red blood cell. Red blood cells are unable to divide, unable to grow, or unable to synthesize proteins. So it's already built in throughout this process of differentiation. Also, red blood cells are worn out in 100 up to 120 days. When red blood cells worn out, red blood cells are eliminated by phagocytize in the spleen or liver. Lost red blood cells are replaced by division of hemocytoblast or hematopoietic stem cell in the red bone marrow. Let's concentrate red blood cell first. So, from stem cell, hemocytoblast divide to form proerythroblast. From proerythroblast, divide to form early erythroblast. This early erythroblast produce ribosome. Ribosome is the site for protein synthesis. From ribosome, hemoglobins start to be synthesized and accumulate. When you look at late erythroblast, hemoglobin is filled in the cell, then nucleus became kicked out of the cell to form reticulocyte. Now this nucleus kicked out of the cell. And reticulocyte does not have a nucleus. Reticulocyte is full filled with hemoglobin. This reticulocyte cannot immediately function as erythrocyte. It needs to mature. So it takes 1 or 2 days for maturation through blood circulation, then eventually resulting in mature erythrocyte. These red blood cells live 100 up to 120 days and disintegrated. Then this hemocytoblast, such as hematopoietic stem cell, continue to generate red blood cell by following this process. Another example is genesis of a platelet. So stem cell, hemocytoblast, or hematopoietic stem cell divide to form megakaryoblast. So if you go back to this cell line, now we are in this line. Just before we saw red blood cell genesis, here's genesis of platelet. This hematopoietic stem cell divide to myeloid stem cell, and myeloid stem cell differentiate into megakaryoblast. Megakaryoblast further divide into promegakaryocyte, and promegakaryocyte differentiate into megakaryocyte. Then cytoplasmic portion of megakaryocyte split up, and each small piece to become platelet. Now this platelet enter bloodstream, and this cell fragment from megakaryocyte break down from megakaryocyte and enter bloodstream and become platelet. And the platelet lives about 10 days. So today you study blood, and you have to know how you categorize blood, liquid portion and cellular portion. Cellular portions can be called formed element, and the formed element can be further categorized: red blood cell, white blood cell, and platelet. And you need to know, when you look at micrographic view, you have to identify what kind of a blood cell the image shows, and then what is main characteristic of each blood cell, and how this blood cell generate, where, from bone marrow, and how this bone marrow, from bone marrow, what kind of a cell line can be formed for generation of a final blood cell. Today we study one of cardiovascular system, blood. Let's solve the quiz question. First, which of these is not a component of plasma? A. Oxygen, B. Sodium ions, C. Platelets, D. Water. You saw this exact same question in the lecture video, so you can find the answer easily. Second question, platelets are derived from megakaryocyte, or platelets are cell fragment, or platelets play an important role in blood clot formation, or ABC, all of them are above are correct. So you can find this answer from lecture video again. This is the example of submitting week 9 assignment. You know my email address, and include your email address and subject Science 101 week 9 assignment, and include your last name followed by your first name in main text body, and write down 2 quiz questions. And the due of this assignment is until December 6th, Wednesday midnight. This is the end of last lecture, blood. Thank you.